Your pancreas makes scissors every single day, sharp enough to cut through meat, an egg, and the proteins of your own cells, and it stores them right inside itself. But if the control system breaks down, the enzymes can switch on too early, damaging cells and triggering inflammation. So how does the body keep these scissors switched off until the last moment? And what emergency fuse saves the gland if one tool switches on too soon? And on top of that, the same gland explains a strange thing: why unsweetened bread can turn sweeter as you chew it. Let's start with the factory of dangerous tools. After a fatty meal, the stomach sends chyme into the gut in small portions, and the pancreas gets to work. It releases bicarbonate, which neutralizes the acid, plus a whole set of enzymes. Lipase breaks down fats. Amylase works on starch, and proteases cut protein molecules apart. But the gland itself is made of cells and proteins. So why don't the enzymes that can destroy meat or an egg start digesting the very organ that made them? The most powerful protein-cutting scissors are produced mainly in an inactive form. Not all enzymes are stored inactive. Amylase and lipase can be released ready to work, but they act on carbs and fats. The main threat to cell proteins comes from proteases. That's why many of them are first built as inactive precursors, zymogens. Trypsin is made as trypsinogen, chymotrypsin as chymotrypsinogen. Until these molecules are activated, their scissors stay closed. But the protection doesn't stop there. The enzymes and their precursors are packed into separate containers, zymogen granules, each one sealed off by a membrane from the rest of the cell. So you get a double defense. The dangerous proteases are still switched off, and they're also isolated in transport containers. When the signal comes, the granules move to the cell surface. Their contents are dumped into a duct and travel through the duct system into the duodenum. And that's exactly where the scissors get switched on. On the surface of the gut cells, there's an enzyme called entropeptidase. It turns part of the trypsinogen into active trypsin. That's the first turn of the key. Then active trypsin starts turning new trypsinogen molecules into trypsin and switching on other proteases. A cascade begins. One activated molecule triggers the next ones, and those keep the process going inside the gut contents. In a few steps, a set of inactive precursors becomes a fully functioning protein digestion system. The gland makes the tools, but the command to switch them all on arrives right next to the food. Biological systems, though, aren't error-free. Sometimes a small amount of trypsin can activate prematurely. For that case, there's one more layer of protection: a trypsin inhibitor, known as SPINK1. It can bind and block part of the trypsin that switched on early, before it sets off a chain reaction. It's like an emergency fuse inside the container, but its capacity is limited. It can't neutralize an unlimited amount of trypsin. If storage, transport, or control break down, the enzymes can activate in the wrong place. Damage cells and trigger inflammation, pancreatitis. It's sometimes loosely called self-digestion, but the real mechanism is more complex. Along with premature activation, there are disrupted calcium signals and mitochondrial function, cellular stress, and an inflammatory response. So the gland isn't protected by one impenetrable shell. Its safety is built from a sequence of barriers: inactive precursors, isolation in granules, export through the ducts, an activation cascade only in the gut, and an inhibitor in case of an error. It doesn't make the proteases safe forever. It keeps them switched off and separates the place they're made from the place they're activated. Now the scissors are on and cutting protein, but right nearby another enzyme is working, pancreatic amylase, which breaks apart the starch from bread, potatoes, and rice. And here a familiar paradox shows up. If you chew plain bread for a while, its taste slowly turns sweeter. Where does the sweetness come from? If we didn't add anything to it. It didn't appear out of thin air. Starch is made up of long chains built from repeating units of glucose. Picture a necklace made of a huge number of identical beads. As long as they're joined into a long chain, the whole thing behaves differently than separate beads. It's a similar story with taste. Free molecules of some sugars taste sweet, while the large structure of starch is barely perceived as sweetness. The breakdown starts right in the mouth. The teeth grind the bread, the tongue mixes it with saliva, and salivary amylase starts snapping the bonds inside the starch chains. But it doesn't take the links off one by one. It acts like scissors, cutting a long necklace into several short fragments. Among them appear maltose, maltotriose, and other small chains. Some of them taste sweeter than the original starch. That's why the taste of bread changes as you chew. But in the mouth, the work is only beginning. After you swallow, amylase keeps acting in the stomach for a while, until the acid penetrates deeper, after which its activity drops sharply. The real continuation is in the small intestine. The acidic chyme mixes with pancreatic bicarbonate. The acidity drops, and pancreatic amylase keeps shortening the chains. But even it doesn't break starch all the way down to single glucose molecules. The final scissors sit on the surface of the gut cells, on the brush border. Here, the short chains meet maltase glucoamylase and sucrase isomaltase, which cut the remaining bonds, including the branch points. Only now are the individual glucose links fully released, and even free glucose is still inside the gut cavity. To get into the body, it has to pass through the gut cell. On the gut side, the transporter SGLT1 grabs it together with sodium and pulls it in. On the other side, the transporter GLUT2 lets it out further, into the blood heading to the liver. Even so, not all of the bread turns into glucose. It has proteins, water, fiber, a bit of fat, and some of the starch may turn out resistant to digestion and pass on through. The speed of breakdown depends on how finely the grain is milled, the cooking, and the amount of fiber. That's why different kinds of bread are digested differently. Bread doesn't turn into sugar by magic. Digestion simply takes apart the glucose chain hidden inside it. What to take away? Different carbs are digested at different speeds. What matters isn't just bread, but which one and with what. And about pancreatitis. It can arise from different causes, including disrupted secretion outflow and toxic damage to cells. It's not just one blown fuse. So the starch is taken apart, and glucose slips easily into the blood. It dissolves well. But protein and fat don't get inside that easily. And protein is tied to the big gym myth. Is it really true that chicken goes straight to your muscles? That's coming up in the next episode. Subscribe to BioCode Lab, where the complex becomes simple.